affects humans in teenage years, and Chelsea is an example of a patient who developed Lafora disease. As you see, she's completely healthy at 14. Chelsea is the young lady of Chelsea's Hope, one of the more organized charities of the human disease based in San Francisco. They pretty much cover the United States. There are charities for the human disease in France, in Italy, in Spain, in Serbia, and there are a whole number of patients in different countries. In the UK, we're aware of about 3 families, but I'm sure there are more. Yeah, so that's Chelsea at age 14. The disease starts with them having myoclonic jerks, generalized seizures, decline in school performance, and all these things gradually get worse. And you know, a picture tells it 1,000 words. This is she from last year, and you can see what she's become, and the video probably tells 1,000 pictures. And this is what she's like at age 22. They go through a period of visual hallucinations, which are always very scary, and these hallucinations are epileptic oftentimes, but also non-epileptic. Then they get into a phase of dementia, which again, it's like everything they get is the worst kind of, like it's the worst kind of hallucination, the worst kind of epilepsy, the worst kind of dementia. They have a disinhibited dementia, meaning they can go and start kissing people and hugging people and doing things that one wouldn't normally do. And eventually they get into a vegetative state and are just convulsing and shaking the whole time until they die from a massive convulsion. This takes about 10 years, during which time their younger siblings are seeing what their sister or brother is going through, and eventually fully aware that they have risk of going through the same. This is EEG. I thought I'd put it in here. I added some slides, Ian, since our conversations yesterday, because some very good questions came up during dinner last night with yourself and Jill. So the EEG, each of these lines, my pointer is not visible recently.
I have a pen with me.
Thank you. So each of the squiggly lines that you see is from a different part of the brain. There are electrodes placed on the brain which measure the brain waves. And you see at age 17 that pattern is more or less normal. Thank you. Oh, it's the screen that makes it invisible. Cool. And so this you can think of as more or less normal, and you see the waves become slower as the child ages, and these spiky things start appearing, which is abnormal, and then these discharges. And when the human patient and the animal is doing a myoclonic jerk, it's because there's this massive electrical discharges going on in the brain. The bigger these waves, the more voltage. This is a voltage measure. So every time the dog or the human is jerking, it's because of these runs of electrical discharges. And it becomes slower and slower, and then more and more discharges. So you see in this page, jerk, 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 lots of jerks. So that's kind of what's going on in the brain while you are seeing the jerks in the dogs. Claire, actually, let's ask Anna, she's a resident. Anna, who is this old man? Can you guess?
Probably is an Italian guy.
He's not Italian.
Okay.
Close.
Probably Mr. Lafora.
No.
No?
No. The old man, the old mustachioed, it's in Spain.
Virtual.
No, Berge was not in Spain, right? Who was in Spain, the big guy in Spain? The teacher of neurology, the inventor of the neuronal basis of brain function.
Cajal?
Yes. Ramon y Cajal, he discovered neurons. And Ramon y Cajal is telling Gonzalo Lafora to go and do great things with his life. And Lafora went and did many great things. He, for example, described the second case of Alzheimer's disease. He worked with Alzheimer at Kaplan, and he also was banished from Spain during the Franco years. He comes from a wealthy family, so he would have been part of the fascist regime then, but he wasn't. He didn't like that. And he was banished to Mexico, where he founded the whole school of neurology in Mexico. And he was very curious to learn many things, so he ended up in this place, which is the Government Hospital for the Insane in Washington, D.C. And there that's what they called such places at the time, and he studied there, and it is in the United States actually that he described these patients with a progressive myoclonic epilepsy, so it's quite striking that they would be put in mental asylums at the time. And the patients would die, and he got their brains, and he had a microscope, but he had no camera attached to the microscope, so he could see, and what he saw was so striking, but he needed to document it, so he drew pictures, because he couldn't take pictures. And he published this in Virchow's archive, and so this is in one of the earliest issues in 1911, and I found a copy in the dungeons of our university library, and they allowed me to take it out, and I scanned it. They had this, what do you call it? They used to put these pictures in the old books, plates or something. And so these are the pictures of Lafora bodies that Lafora saw in these patients' brains. So this, for example, is a pyramidal neuron. This is its dendrite. This is its nucleus, and this is the Lafora body. A huge body taking up much of the cell, and also found in the dendrites, but never found in the axons. And they come in all sorts of shapes, which he drew. And now I will show you the real-life current examples. So this Lafora body is so large that it outgrew the cell it was in, destroyed the cell, and is freely floating in the neuropile, meaning in the brain. And here's one that is so big that it's occupying this whole cell body. You can recognize the neuron and inside the Lafora body. And there are many others of different sizes. This is a typical juxtanuclear Lafora body, meaning they for some reason are close to the nucleus of the cell. This is one in a dendrite. This is an axon. You can recognize it by the synaptic vesicles. And this is the dendrite. And the dendrite is filled with this Lafora body, which you can see are fibrils. There are masses of these fibrils. And the next question is, what are those fibrils? Because the answer to what those are and how they form is the answer to Lafora disease. That's what's abnormal in these patients and dogs. So they are fibrils which were resolved biochemically and shown to be chains of glucose. Glucose is the small sugar molecule. And so they are chains of glucose. And usually when you have a chain of glucose that is 8 units or longer, it tends to become insoluble and precipitates. So if you have a solution, let's say, if you put sugar in here, if it's the usual sugar we eat, it's small, it's 2 units. It will stay dissolved. But if it's longer, it will start precipitating and accumulating in the bottom. and then accumulating over time, and it will be a big mass in the bottom here. That's like starch. The potato cell or the rice cell and so on have starch, which is chains of glucose like this, which are supposed to precipitate and accumulate so that we make bread from it or rice and we eat it and it's fine. Except that's not supposed to be the case in a mammalian cell. So we're not supposed to have this kind of thing. Instead, in our cells, the chains of glucose is because glucose is our energy source, and to store it we have to put it in chains. So in starch, in plants, it's like that. In humans and dogs and other mammals, it's like this. It's spherical. So what happens is the chains are hidden inside the sphere because of all this branching, and the ends of chains are exposed to the outside. So it makes a ball which has 50,000 glucose units in it. It's massive. Yet it remains soluble because the surfaces are not exposed to the outside. It's the largest molecule in the cytosol. The only thing that's larger than this is DNA itself. And so this stays soluble by this fantastic biological state. So the mystery of Lafora disease is how come this becomes this? That's the question we're trying to solve. If we solve that, we understand Lafora disease. So one way to understand a genetic disease is to find the genes that cause said disease. So we found the genes, this one in 1998, EPM2A, we called it Laforan in honor of Lafora. And this gene turned out to have a carbohydrate binding domain, which isn't surprising. This is the protein that is made by this gene. And that's not surprising, obviously, because the protein has to act on carbohydrate. And it has a phosphatase domain, which already gave us a clue of what it does. It would be removing a phosphate group from whatever it binds to. And eventually we figured out that it's removing phosphate from glycogen itself, and I'll come back to that. Then, when we found this gene, this explained half of our cases. The other 50% of cases did not have mutations in this gene. So we needed to find a second gene, and that proved difficult in humans. And it was around that time we were aware that there are quite a few publications of Lafora disease in dogs. And as I was wondering who to contact to see if dogs can help us, I got an email from Claire. And, you know, to make a long story short, through the dogs and the Dachshund clubs and owners and everybody who helped us, we were able to find the second gene, which we call EPM2B, and which we named Malin, out of, as kind of another bit of information. The reason this is called Malin is based on one of the families with Lafora disease in Quebec. Quebec is a province of Canada, which is a French-speaking province, and there are, I think, 8 million Quebecers now. They all descend from about 1,000 or a few thousand immigrants who came there, I can't remember in which century, very early on in the founding of Canada, and all of them descend from that small group of people. And they have kept very good church records. And because they all descend from a small group of people, they have a number of inherited diseases by what's called the founder effect. This is very similar to the canine diseases. If you have a founder and that founder has lots of children and then their kids marry each other, then you get diseases. And their last name was Malenfant. Malenfant means sick child, possibly because the disease has appeared in this family. You know, I'm not sure that is why they're named like that, but they are named sick child. So she was so helpful to us. Both her children died in the process of her helping us, but we named this protein. Also, mal in French means seizure. Grand mal, petit mal, you may have heard. Okay, now the next slide is from questions that came up last night, Ian. And Ian and Jill were asking about the mutations in Lafora disease in humans as compared to dogs. So in humans, this is the situation. This is that first gene, Lafora, and the mutations can be anywhere in the gene. The longer these bars, meaning more patients have that mutation. So this particular mutation, the largest number of patients have this mutation. But the point is that mutations are found everywhere else as well. So you can, and this is common in any disease, you can mutate the protein at any of its points and it won't work well. So you get the disease. Okay? Very important to keep in mind that there are mutations throughout the gene in humans. This is the second gene, the malin one. Likewise, mutations found throughout the gene, which is not the case in the dog situation. So now we're going to go to the dog. This is work done by Hannes Lohi in my lab 13 years ago or so when we started working on the canine world. Hannes is now a big professor of genomics of canine diseases, and Saya was his PhD student and now is in my lab. So it's very nice, the second generation from Hannes. So in dog, the dog genome has a particularity that is only found in dogs and not in any other species. Not in humans, not in cats, not in bears, only in dogs. Inside the gene, this is the malin gene, inside the gene there is this sequence of nucleotides, you see GCCL, which is repeated twice. This T, this one is again a similar sequence, but there's just a T in there. You can disregard this T. In any dog of any breed, you have these two copies of this sequence. Some dogs have two, some dogs have three. So you can imagine another bit of this D placed in there, just a bit longer. So 2 and 3 is normal. That's a normal dog. What happens in the dogs with Lafora disease, this repetitive sequence, every once in a while, expands. So instead of having 2 copies, you have 20, 30 copies. So suddenly the gene is, that's 20 times 12 because there's 12 here. It's hundreds of nucleotides longer. And because it's so big now and abnormal, it cannot make protein. And so there's no myelin protein, and so the dog has Lafora disease. If we look at other species, the bear, the American bear, which is a close relative of dogs evolutionarily, has only 1 copy of this, does not have the second copy. The cat has 1 copy, but already it's different. There's an A in here. The mouse has various differences. The rat is similar to the mouse, unsurprisingly. Sus is pig, I think. The pig is a pig. And the human is different from the dog. And this is monkey. It's kind of similar to the human. So none of these species can have the canine mutation because they don't have these repeats. They cannot have this expansion mutation. This expansion mutation is unique to dogs. Now, Next, I will talk about the testing, and again, this question came up yesterday. The testing we used to do and the testing we do now, and the problems with the old test compared to the new test. So, in the modern era of genetics, every old tests are based on PCR, polymerase chain reaction. This was invented some years ago and won the Nobel Prize. What PCR is it's complicated, but basically the system starts making copies of your target sequence. So let's say you want to test this gene, so you make copies of it, hundreds and thousands of copies. So you have your whole genome, but you focus on the part that you want to sequence, and you make thousands of copies of the part you're focused in. And now, because you have thousands of copies of that and only one copy of everything else, you can see that bit much, much better. But the PCR enzymes have to be able to sequence through this. And by definition, this mutation, the reason it's a mutation is because enzymes have a very hard time going through this expanded repeat. So if you do a normal PCR on the expanded allele, it doesn't work. You can't expand anything. So in the old test, if you do the PCR and get a blank, we could say this dog is effective, because we can't sequence through it. And if we are able to get a band, we say it's ineffective. So that part is easy. But what about the carrier? That's the difficulty. So Hannes came up with a system by deamination and so on. It's complicated to explain, where he made the PCR work. And so the wild type, the normal, is this size, which is the normal size. The affected dog, this is a miniature wire-haired Dachshund who has the disease, will have a large size. And the carriers would have a small and a big. But look at the quality of the PCR reaction. For the small, you get a nice thick obvious band. For the big, it's very faint. And sometimes, like in this example, it's so faint that you can barely see it. I mean, it's there here. If you come close, you will see it. But sometimes you can't even see it. And so the difficulty with the carrier testing using PCR is that there always is a small doubt about whether you have a result that's more like this or more like this. You can have a carrier who will look like their wild type, and you wouldn't know. Now this is done from DNA from blood, and there is much, much more DNA from blood than there is from saliva. From saliva, there is very little DNA. The PCR usually works on saliva, but in this case, it's much, much harder because there's so little. And so you will end up with situations where you can't amplify this, and you will misdiagnose a carrier as unaffected. This is the difficulty with PCR-based tests. It's possible to overcome, as you see, we knew how to overcome it in 2005 when we published this, but always left with question marks. So instead of doing this, we went back to a very old technique before the invention of PCR. Before the invention of PCR, people did other things, such as something called Southern blot. And Dr. Southern is someone
But my father knew.
that Claire's father happened to know. So in Southern blot testing, you don't amplify anything. So you don't have to sequence through this. You take it as it is. And you just cut it, and using a radioactive probe, you can see and ask, is it big or small? Because you don't have to do anything to it, it's in its natural state, and you cannot go wrong. It's impossible to make the mistake in the Southern blot that we can make in the PCR-based test. So you see the small one, which this is a wild type, Dachshund normal. This is a sick dog, and this is a carrier dog. So it's very, very straightforward and easy. So that's about the testing. The next slide is going back a little bit on the evolution of this repeat, and I put it here to make a very important point for the Kennel Club itself. So, in evolution, 60 million years ago, the cat creatures of the time and the dog-like creatures of the time were one, and then they diverged. So you get the Felidae and the Canoidae. The Felidae are here. By the way, all these different species, Hannes went to the Royal Ontario Museum, and they had this Finn coming into the museum and saying, I want pieces of tissue from all these species that they have preciously kept. I thought, there's no way they're going to give you anything. But he came back with all of them. He was just amazing that So, here are the cats. The regular cat, lion, tiger, leopard, snow leopard, cheetah, lynx. And all of them don't have that extra copy of the repeat. And they have an A here in this sequence. Now, the Canoidae, 50 million years ago, split into the dogs, the Canidae, and the bears, Arctoidae. Now, the Canidae, the Canis familiaris is the regular dog. Then you have, for example, the dingo, that Australian dog-like creature. The gray wolf, the red wolf, the coyote, the jackal, the dole, and the gray fox, and some other fox. All of them are like the dog. Well, sorry, not all of them. Some of them have, let's see, except for this fox, actually, all of the other ones have the repeat. You see? The bears, which branched out from the dogs, don't. They have only one copy. So you see that the bears are similar to the dogs, but they don't have the extra copy. And so the skunk, the black bear, the polar bear, the brown bear, the grizzly, the raccoon is until the badger. And I learned yesterday that our Dachshunds will chase the badger. And the poor badger gets chased by the dog, but in terms of this particular disease, the badger is safer. Now, my point from this is that Lafora disease is not a disease of humans that happens to present in dogs. Lafora disease is a disease of dogs that happens to present in humans. Because dogs are predisposed to have Lafora disease. Don't get me wrong though, this is not a big predisposition, otherwise lots and lots of dogs would have it. Relatively speaking, dogs have this small defect, particularity, whatever you can call it, which leads them occasionally to go from this double sequence to this large sequence. And this happens only in dogs and not in bears and cats and mice and humans. So Lafora disease is a canine disease which happens to also occur in humans. To just make this point a little bit stronger, every species has particularities like this. In humans, there's another disease which is particular to humans and will never occur in dogs. It's very similar. It's called Unrecht-Lundborg disease. It is a myoclonic epilepsy similar to Lafora disease. In this case, this is a different gene called the cystatin B gene. In this case, the humans happen to have this repetitive sequence. And it is the humans which go from a normal sequence. You see 3 copies here, 1, 2, 3, to these big expansions, in this case more than 50 copies. This disease, which is also a bad disease, never occurs in dogs and cannot occur in dogs because they don't have this particularity. Whereas Lafora disease occurs in dogs, and occurs in humans, but much less in humans than it would occur in dogs. So I think this is important for the canine world to be aware of generally, because this disease happens not just in the miniature wirehaired dachshunds. With Claire, we've seen it in basset hounds, in beagles, and there are reports of it in several other breeds. And I think the more you look, the more you'll find it. I think many epilepsy cases in dogs may well have this. Dogs have other epilepsies too, but they could have this disease. You don't need to follow this. This is just up there to say that part of our lab is dedicated to answer the question why glycogen becomes starch-like in Lafora. And so we have many complex experiments going on to try to answer that question. But we couldn't just wait to figure out this complex problem. And if we had a way to treat our patients without having full knowledge on the pathogenesis of the disease, we really wanted that. And that's what leads me to the therapy ideas. So again, we want to know how this becomes this, and half the lab is focused on that. But then we ask the question, one day we had this kind of brilliant moment, and it's funny that, this idea could have occurred to us much earlier. It's easy to say now in hindsight. Everything is easy in hindsight. But anyway, it happened one day that we thought, okay, so these abnormal chains are glucoses put together. And there's only one enzyme in mammals that can do that, put the chains together, and it's called glycogen synthase. What if we decrease glycogen synthase? Would this reduce this and fix the problem? So we decided to do that experiment. So we got mice that don't have glycogen synthase, so they can't put these glucoses together, and are mice with Lafora disease. And we bred them together and ended up with mice that have Lafora disease but who don't have this enzyme. And so, to our great surprise and happiness, these Lafora bodies, as we said, are accumulations of these abnormal glycogen. They went down to normal. This is the double knockout, meaning a Lafora disease mouse who does not have the glycogen synthase enzyme. And so suddenly they become like the normal in different tissues. Saya, maybe you can press this button so we don't go back and forth. This is skeletal muscle, and each of these is a muscle fiber. And all this pink stuff is Lafora bodies. So in these mice that cannot make Lafora bodies, don't have Lafora bodies at all. Even at high magnification, there's absolutely none. Next. This is the brain. This is the cortex of a mouse with Lafora disease, and all the pink stuff is Lafora bodies. And the dog's brain would look like this. And the treated mouse, meaning the mouse that doesn't have glycogen synthase, has barely any left. This is a different region of the brain, the hippocampus, and you see all the Lafora bodies. They are completely gone. Next. Lafora disease is a neurodegenerative disease, so these Lafora bodies, as we saw in one of the first slides, destroy eventually the cells that they're in, and the neurons get lost. And the lost neurons get replaced by glia, which are the scar cells of the brain. And you can check the amount of scarring by using a stain that tests them specifically. This is the hippocampus, and all this black is GFAP, which is the stain of scars. And you see it's all scarred because the brain cells have been lost. Whereas the animal that was treated with or that was bred with the one that doesn't make glycogen synthase doesn't have the scar. Next. This is the myoclonus counts. So the mice are not a very good model for Lafora disease because they have the pathology which you saw, but they only live about 3 years maximum, and their epilepsy is much milder, so I just want to make the point that the dog, like I saw again yesterday and I had seen before, is looks much more like a human patient in terms of the amount of myoclonus and seizures and so on. The mouse is a good model on the pathology level, but not a great model at the clinical level. Nevertheless, the mouse does have extra myoclonus, these jerks, and so that myoclonus goes away with the treatment. Next slide. So, our goal is to reduce glycogen synthase. You can press again, please. If we can reduce glycogen synthase, we know that we will have a cure or a therapy for the disease. Next slide. So glycogen synthase is a protein. The proteins are made, they have a gene that makes a messenger RNA, which makes a protein. So we can bring this down by targeting protein, RNA, or gene. And so we have experiments going on in the lab at all these levels. We're using CRISPR technology to knock out the gene. We're looking for small molecules that would inhibit the protein. The advantage of small molecules being you can take them back out. And we're targeting the RNA. We're doing all this work in parallel. The one that has taken the lead right now is the work that Saya did against the RNA. And she'll talk about that. So she will explain the kind of molecule we use to bring the RNA down and thus bring the glycogen synthase down. Next slide. This is Saya's slide. Go ahead. This is a recent picture of our group. There's a few people missing. Felix is doing all the, Felix and Sylvie and others are doing all the work to try to understand how Lafora bodies happen. And then others are doing all the therapy work, Aaron, Staya, and so on. And some people do some of both. This is Lafora. These are some children with Lafora. I don't remember anymore who's alive and who's dead. But these are, our patients that we're trying to help. So again, we know that by simply reducing glycogen synthesis, we can fix this problem, irrespective of understanding precisely why it happens. So we have kind of circumvented the unknown bit towards a therapy, and Saya will explain the therapy. Any burning questions right now?